Hello and welcome. Let's talk today about the nervous system and nervous tissue. When we talk about the nervous system is considered the master controlling and communicating system of the body. Now, we'll visit first a few functions and then we'll progress further into the nervous system. Now, when we talk functions of the nervous system, it's got three basic functions, and these three basic functions will include number one, sensory input. When we talk about sensory input is one function where the nervous system uses its millions of sensory receptors to monitor changes inside and outside the body. The second function then is integration. The nervous system, it processes and it interprets sensory input and then decides what to do with it. For example, there. Third then is motor output. When we talk motor output, the nervous system, it causes a response by activating effector organs. When we say effector organs, we're talking muscles and we're talking glands. So here we could see sensory or afferent neurons are going to be responsible for conducting signals from our receptors to the central nervous system, the brain and the spinal cord, where then the interneurons are association neurons, which are confined in the CNS, will be responsible for integrating that information. And then they'll dictate out a motor response via our motor or efferent neurons, which then we said conduct impulses to our effectors, such as muscles and glands. Another example there. Patient becomes thirsty, they see a glass of water. Integration activates those certain muscles. Which then are going to be, so we can see the same thing here when we talk about the same three basic functions: sensory input, integration, and motor output. Patient is thirsty. They see a glass of water with their sensory receptors found located inside the eye. The brain says, all right, well, let's activate the muscles that can pick up that glass that's there and let's drink it. Next, let's move to the organization. When we talk about the organization of the nervous system, the division of the nervous system, we'll see the nervous system gets divided into the central nervous system, the CNS, and the peripheral nervous system, or the PNS. Let's talk first CNS, and then we'll move over to the PNS. Now, when we talk CNS, our central nervous system, we'll see our central nervous system is going to be made up of the brain and the spinal cord. It will be made up of the brain and the spinal cord. Now, when we talk about this central nervous system, we can see that organization here as well. And we see that same organization here also. I like to use this picture for the best illustrative purpose. So we'll go from this picture. Now, when we talk about the brain and the spinal cord, if you recall, the brain and spinal cord are going to be found in the dorsal body cavity. Now, when we talk about this central nervous system, the central nervous system is going to be the integrating and command center of this central nervous system, of the nervous system. So it is the integrating and command center of the nervous system. It receives sensory input and it dictates a motor response, as we described. Now, when we talk about the peripheral nervous system, or the PNS, the peripheral nervous system is the nervous system outside the CNS. So everything outside the central nervous system. which is mainly nerves, which are bundles of axons that are going to be found extending from the brain and spinal cord. We'll see we have spinal nerves and cranial nerves that we're going to go through and we're going to check out and you'll be responsible for knowing about. Now, when we talk about this peripheral nervous system we can see is further subdivided. It gets further subdivided into the sensory or the afferent division and the motor or efferent division. So when we talk about the sensory or the afferent division, here we'll see the nerve, it's made up of nerve fibers, and these nerve fibers here are going to be responsible for conveying impulses to the central nervous system from the sensory receptors located throughout the body. Now, this division is made up of two fiber types. The first fiber type here are going to be your somatic or your afferent fibers. And second, you have your visceral afferent fibers. So first, when we talk somatic afferent fibers, these somatic afferent fibers, they convey impulses from your skin, skeletal muscle, and also our joints. Now, the visceral afferent fibers transmit impulses from our visceral organs, the organs found in the ventral body cavity. Now, the second division we have here, our motor or efferent division, it transmits impulses from the CNS to the effector organs, to the muscles and glands. And it gets further subdivided into the somatic, or what we refer to as the voluntary nervous system, and into the autonomic, or involuntary nervous system. So when we talk somatic or voluntary nervous system, you'll see the somatic or voluntary nervous system, it has somatic nerve fibers. And these somatic nerve fibers, they conduct impulses from the central nervous system to your skeletal muscle. Then the autonomic or involuntary nervous system, it's going to consist of visceral motor nerve fibers. And it's going to regulate the activity of smooth muscle, cardiac muscle, and our glands. Now, this autonomic nervous system, we cannot control. Now, when we talk about this autonomic nervous system, you can see it gets further subdivided. It's going to get further subdivided into the sympathetic division and parasympathetic division. Now, when we talk sympathetic division is going to also be known as the fight or flight division. And then the parasympathetic division is also known as the rest and digest division. Now, when we go through and we talk about the sympathetic division, I want you to know, it mobilizes our body's systems. It will mobilize our body systems during activity or under stress. It mobilizes, you can say, our body's resources or our body's systems during activity or under stress. Now, when the sympathetic division is activated, it mobilizes body systems during activity. So if we talk about the heart, for example, it will increase heart rate. If we talk about the lungs and we talk about basically the bronchi, it'll cause bronchodilation to allow more air to get in, right? Under periods of stress or activity. So if you're doing some type of activity or you're under a period of stress, you want to ensure your heart rate is high, so that way blood is going everywhere it needs to go. You're getting enough oxygen in, right? We talk about the eyes. The eyes you're going to have, you'll see a dilation take place. Pupils will dilate to allow more light in. So in case you need to, we said fight or flight. When we talk about the circulation making its way to the digestive tract, that circulation is going to be basically minimized. And all that blood that's supposed to be going there is going to be rerouted to our basically skeletal muscle in case, again, we need to fight or flight. So you could see there, when we go through and we talk organ by organ, you've got specific changes that are going to be taking place. Then when we talk about rest and digest, or this parasympathetic division, when we talk about this parasympathetic division, it conserves energy. It conserves energy and it promotes housekeeping functions at rest. It conserves energy and it promotes housekeeping functions at rest. Activities basically that occur at rest, such as digestion, or right, we said rest and digest. So this is where we would be resting and digesting. Now, here, let's go back and revisit the same examples with the heart. When you have parasympathetic division activation, heart rate is not going to be increased. It will go back to being what it was at rest. When you talk pupils, you won't have that dilation, you have a constriction. You're resting and digesting. When you talk about the lungs, you won't have that bronchodilation as you had with sympathetic activation. So that'll go back to what it was during resting. So you see all these changes that take place whenever one system or the other is activated, and you're always going to be in one state or the other. Next then, the cells of nervous tissue. Cells of nervous tissue. Now, when we talk about the cells of the nervous tissue, here we will check out neuroglia and our neurons. So here we've got a nice table listing all of them. And then here we've got a nice illustration depicting all of them. And then here we'll go through each of them then one by one. Now, when we go through and we talk about our neuroglia are also known as our glial cells. Also known as our glial cells. We have 6 types. We have 4 in the central nervous system, and we have 2 in the peripheral nervous system. Now, when we talk about neuroglia of the CNS, here's the first cell we'll talk about in relation to the CNS, and this cell is your astrocyte. When we talk about astrocytes are the most abundant and most versatile glial cell. They are star-shaped, branching sea anemones is how they're described. I think they all look like sea anemones. Now let's talk about their function. So here you can see they have star-shaped, we said, right? Because you can see all these processes that are radiating from that cell body where the nucleus is found. These processes, what they do is basically they are going to, number one, support and brace and anchor our neurons. They'll support, brace, and anchor our neurons to their nutrient supply lines. Now here, with them clinging tightly to the nutrient supply lines, this is going to then allow them to receive all the nutrients that they need. They can extract basically out whatever they need to send over to the neurons. Now, these astrocytes, they also control the chemical environment around the neurons. Second, what they do is control the chemical environment around the neuron. So here what they do is they mop up any leaked potassium ions, and they recapture and recycle released neurotransmitters. And then third, they're going to influence neuronal function. They participate in information processing in the brain. The second cell type I'd like you to know about then is the microglial cells. These microglial cells are small, ovoid cells with long, thorny processes. Now, the function of these cells is to monitor the neuron's health, and they also function as macrophages, where they have the ability to phagocytize dead material, microorganisms, or dead neurons, or neuronal debris. Next, then, we have our ependymal cells. Our ependymal cells, their structure is going to range in shape from squamous to columnar, and many are ciliated. And when you talk about their function, they're going to function by lining the central cavities of the brain and spinal cord. When we talk lining the central cavities, we're talking about the ventricles. They line the ventricles of the brain and the spinal cord, where they form a permeable barrier between cerebral spinal fluid that's going to fill those cavities, as we see here on one side, and the fluid, so this fluid, and basically this tissue, this CNS tissue. So they form a permeable barrier between cerebral spinal fluid that fills those ventricles, and then the tissue that's basically found making up the CNS. And the cilia that we see here, it's going to help to circulate that CSF. Next then are our oligodendrocytes. Now when we talk about our oligodendrocytes, their structure is, they look similar to astrocytes, however they have fewer processes. Now when we talk about oligodendrocytes, their function is that they produce myelin in the CNS. They produce myelin in the CNS. Next, then, let's talk about the neuroglia of the PNS. When we talk about the neuroglia of the PNS, here we have first our satellite cells. So 4 you saw in the CNS, 2 in the PNS. Satellite cells and Schwann cells. Now, satellite cells, they're found located surrounding the neuron cell bodies, located in the PNS. Their function is the same as astrocytes. Then the Schwann cells we see here, they're found located surrounding the nerve fibers. Found surrounding the nerve fibers in the PNS. Now when you talk about the function of these Schwann cells is that they produce myelin for the PNS. They produce myelin for the PNS. Then the next cell then we have here is our VIP cell, the neuron. Now when we talk about our neurons, here's a couple of different images, so we'll use this image, it's a little bit more simpler. Our neurons, first let's talk characteristics. Longevity. They have longevity as long as they receive good nutrition. So with good nutrition, they can function for a lifetime. Right? Your neurons, we don't get a transplant at the age of 50 or 40 or anything like that. They last a lifetime. They're amitotic. They lose the ability to divide and cannot be replaced if destroyed. Stem cell research hopefully is going to help with things like this. Excuse me. Metabolic rate next. They have a very high metabolic rate. They require high oxygen and glucose. Let's go through the different regions of the neuron. First, we have here what we call the cell body. The cell body is also known as the soma. And it consists, you can see, of a spherical nucleus. A nice spherical nucleus. A conspicuous nucleolus that are both surrounded then by cytoplasm. The nucleus, right, contains a nucleolus. Along with that, genetic information. The nucleolus is responsible for producing rRNA, as you've learned. So then here we have cytoplasm. Basically surrounding those structures. Next, then here we can see the function. Let's talk about the function. The function of the cell body, or the function of the soma, is that it is the major biosynthetic center of the neuron. It contains the usual organelles. So when we say it's the major biosynthetic center, here that means it's responsible for producing proteins and various types of neurotransmitters. So the nucleus contains a nucleolus, we've talked about there, and genetic information and all of that. Here then we have what we call Nissl bodies. Nissl bodies are basically the rough ER, the rough endoplasmic reticulum. And what they do is they produce proteins, as we mentioned. Then we have in here also neurofibrils. Neurofibrils are neurofilaments, which are important in maintaining cell shape and integrity. Next, then, let's classify our cell bodies. When we classify our cell bodies, they can be classified as either nuclei or ganglia. Now, when we talk nuclei, when we use the term nuclei, we're talking about clusters, collections of cell bodies in the CNS. Now, when we use the term ganglia, we're talking about clusters of cell bodies in the PNS. Next, then, let's talk about the various processes. Let's talk about the processes. When we talk about processes, we're going to be talking about extensions found from the cell body of neurons. Now, here we have first dendrites we'll talk about. When we talk dendrites, their function is that they are the main receptive or input regions. They provide an enormous surface area for receiving signals. When you talk about their structure, they're short, they're tapering, and you can see very diffusely branching extensions. Like we said, they're the main receptive or input region. They receive the incoming signals. This soma, the cell body, can also receive incoming signals as well. It can serve as basically the region that receives incoming signals too. Next, then we have the axon. When we talk about the axon is basically any long, you can see, process coming off of that cell body. We can also refer to it as, we said, the nerve fiber. Now, the function of the axon is it's the conducting region. It will generate here at the axon hillock, and then it will transmit. Nerve impulses, electrical impulses. Now, when you talk about the structure of the axon, here you have first the axon hillock. This is the cone-shaped initial region of the axon. It is the cone-shaped initial region of the axon, and this is where the action potential starts. You've got a region inside called the trigger zone, and that's where you'll see the action potential starts. Next in here we can appreciate the Schwann cells and how they're producing myelin. So when we talked first, the oligodendrocytes, we saw here they produced myelin in the CNS, we said. So their function was to produce myelin in the CNS, and you can see one oligodendrocyte produces myelin for many nerve fibers. But when we talked about Schwann cells, which also produce myelin, however, in the PNS, you can see many Schwann cells are required to produce myelin for one nerve fiber. Many Schwann cells. So this is one Schwann cell, produces the myelin in that region. Another Schwann cell produces myelin for that region. Another Schwann cell produces myelin for that region. So here you can see then, when they produce myelin, as you can see from this view as well, so here we've got the Schwann cells. They produce the myelin sheath. So here you can see the Schwann cell is going to basically encircle one segment. And once it encircles the one segment, then it continues to produce these concentric circles, wrapping around. Basically, you can see the plasma membrane. And then finally, completing myelination, where you have then this outer husk, the neurolemma formed. And then you can see the myelin sheath right underneath. And then here's the nerve fiber. The myelin sheath is a whitish, fatty, segmented nerve fiber covering. And the neurolemma, we said, is the outer husk, the outermost layer, basically. Now, what does this myelin sheath do? It helps to conduct nerve impulses faster. Helps to conduct nerve impulses faster. So if you were to compare what we're seeing here, this is not a complete myelination, right? Here we have unmyelinated regions, which we said are called the nodes of Ranvier. Here what happens is, we also spoke of, that action potential gets regenerated. So if we were to compare this, the speed from this nerve fiber, to one that's fully myelinated, the one that's fully myelinated, its potential is going to be traveling faster. And then one that's non-myelinated, zero myelin, and we compare that to this one here, obviously, this one is going to be faster than the one that's non-myelinated. So you can see we can have various types of nerve fibers depending on central nervous system, peripheral nervous system, or where we're at, whether they're myelinated or even non-myelinated. Next then, when we talk myelinated versus non-myelinated, we're getting into white matter and gray matter. White matter is regions of the brain and spinal cord that contains dense collections of myelinated fibers. We talk gray matter contains mostly nerve cell bodies and unmyelinated fibers. So let's scoot along the neuron. And here then we can see that axon is going to split, and it splits into the terminal branches. Splits into the terminal branches. The terminal branches are branches or extensions from the opposite end of the cell body, from the opposite end of the axon. These terminal branches then are going to end. They're going to end as the terminal boutons or the axon terminals we see here. And that's the secretory region. That's where they're going to secrete from their neurotransmitters. So here what we could do is let's magnify that region there. And when we magnify that region there, you can see what we have is we'll have something that looks like this. Here's the axon terminals. And then here's the branches. And we'll grab one of these branches. And here we'll see this is what it actually will look like. Kind of enlarges out like that. And inside of here, we're going to have vesicles. And these vesicles are filled with neurotransmitter, right? We saw if we're talking muscle, skeletal muscle, that neurotransmitter was acetylcholine. So it depends on where you're at, what you're talking about. Now, this region in here, this is what it looks like. So that's the axon terminals. They're filled with, first let me tell you, the axon terminals are knob-like distal endings of the terminal branches, and they're filled with neurotransmitters that are found within vesicles. Next then we have the term synapse. Synapse is going to be a junction that mediates information transfer from one neuron to the next, or from one neuron to an effector cell. Basically saying here, if we have another neuron, let's say, or a muscle cell, okay, let's draw a muscle cell, and we'll put some striations in there, right, and so forth. Now, here is basically the synapse now. So again, it's the junction that mediates information transfer from one cell over to the next. So that's what we have here. A junction where two things come together. So right here we call it a synapse. All right, next then let's talk about classification of neurons. Now we can classify our neurons by structure or by function. So here we can see they are classified by structure. We structurally classify our neurons as multipolar neurons, which is a typical neuron that we've been talking about. Multipolar because they've got, you can see multiple dendrites, multiple processes basically, multiple dendrites, and one axon. So many processes extending. Now our bipolar neurons have two processes extending from the cell body, a dendrite and an axon. Unipolar, only one process, and that one process is an axon. So according to structure. So some various types of neurons according to structure. And then also according to function. When we talk according to function, we saw that here then in the beginning, where we have basically sensory neurons, interneurons, and then motor or efferent neurons. So sensory or afferent, interneurons or association neurons, and then motor or efferent neurons. And last, what we have here then is the classification of these processes. Now, which process we're going to be classifying, it's going to be, you'll see the axons. Now, when we classify these axons, they get classified according to two different terms, as either tracts or as nerves. Now, we've already established the term nerves, we said, are bundles of axons in the PNS now. I want you to add there, in the PNS. Last then are tracts. They're bundles of neuron axons, right? Neuron processes in the CNS. In the CNS. We have various tracts in the CNS. They're responsible for controlling various functions in the body as well. And these tracts can get very specific to where they're going to be controlling specific functions in specific areas of the body. It's a very interesting subject as well.